Hello friends, in this tutorial we will talk about applications of AI in pharmaceutical sciences. So we will talk about what are the processes or what are the steps for making a machine learning model for drug discovery in general, or for that matter whatever is the purpose using chemicals for machine learning. What are the usual steps and data pre-processing steps in machine learning based drug discovery projects? So without much ado, let me go ahead. So we all know, as already I have told in my earlier video, machine learning means 80% data handling and only 20% is the fine tuning of the machine learning model. So what is the meaning of data handling? To collect the data, to pre-process those data, to remove unwanted data, and then to train, test, and validate machine learning model using fine-tuned, highly filtered, genuine, usable data. So first step is dataset collection and filtration. A dataset here, dataset means dataset either on paid basis or public basis, which can be used for training the machine learning model. The input data should be of similar type for which we are planning to use machine learning model. A data set of various molecules for which inhibitory activity against target protein is available will be downloaded from binding DS database and Campbell or Chambell database. For example, if we are planning to find a drug molecule for treatment of cancer, then we have to find the cancer inhibitors, inhibitor drug molecule or the compound which are able to inhibit the receptor or the protein responsible for cancer, that is the cancer. So a data set of various molecules for which inhibitory activity, their activity must be available, only those molecule will be extracted or obtained from the database. Like Campbell database will give data of chemical, and binding DS will give you the binding affinity or the score of after binding of the ligand, that is drug, with the protein, that is receptor. Only those molecules whose activities are given in terms of IC50 or EC50 should be selected. The collected data should be subjected to refinement in order to remove duplicate data, because you cannot rely on those database. You have to remove the unwanted compound, duplicate compound, missing data values, and other type of misleading data or outlier should be removed or filtered or discarded from you by you. So you have to remove duplicate molecule, salt forms, heavy metals, means you have to remove garbage from your data input, because garbage in, garbage out. Whatever data you are using to train machine learning model, it will show the same type of output, which will not be useful. Further, we have to use strict quality control filter like IC50, EC50, uniqueness, SMILE, assay. SMILE means valid SMILE. SMILE is a way to represent it is a way to represent chemical or the drug in front of machine learning model in a SMILE pattern. Assay means the value of biological assay in terms of IC50 or EC50. So then finally we will obtain certain filtered unique inhibitors with the respective half maximal inhibitory concentration. For class labeling, IC50 cutoff value of 1000 nanometer will be decided. So you should have two things, X and Y. X means independent feature or your input. Y means output or label or your result. So here X is independent feature, that is various type of drug molecule or the compound, and Y will be their inhibitory potential, that is label. So what is IC50? It is label. So molecules having IC50 less than or equal to 1000 nanometer will be selected. So there are a huge number of molecule. You should get only some filtered molecule. So you have to apply various filtering criteria. The one criteria is, okay, we will take only those molecule whose Y value or whose label value is equal to 1000 nanometer. We will remove other all material from our training or testing data set. So molecules which are having IC50 greater than 1000 nanometer will be selected as negative or non-inhibitors. So we will not choose those molecule as positive. We will show that if molecule has IC50 more than 1000, they will be negative. So you should have both type of data, positive and negative, for training the machine learning model. If you only show positive molecule, then how machine learning or deep learning model will learn to discard or to identify negative values or unwanted label or unwanted compound. In this way, further molecules will be filtered, for instance, out of x minus 500. So you will keep on eliminating unwanted molecule using various filtration or the eliminating criterias. IC50 or AC50 values will be converted into negative logarithmic of half maximal inhibitory concentration, where IC50 is in form of dimensionless activity that can be approximated numerically as molar concentration. The higher PIC50 indicates exponentially greater potency. The PIC50 is used for the designing if various regression based predictions algorithms are to be used. We all know classification is a discrete phenomena where you try to classify or put things into two categories, 1 or 0, active, non-active, dog, cat, sort of things. In case of regression, it is a continuous variable and it tries to fit the data between a straight line. Now the next step is data preparation. The chemical name will be used to extract the chemical information like Smiles, which was then converted to 3D SDF file using OpenBabel. It is OpenBabel is software which can be used to convert molecule or Smiles into 3D structures of the data. Descriptor generation. Molecular descriptors are used to represent the physicochemical properties of molecule, which are mainly used in SAR and QSR studies. Total number of descriptors, because ultimately what are the inputs? Inputs are the material or the data set with some features. So in case of molecule or the chemical compound or the drugs, the features are various descriptors: number of bond, type of bond, then bond feature matrix, number of nodes, number of bonds or edges. These are the descriptors or the features which are responsible for making one particular molecule that particular molecule. Total number of descriptors will be calculated using PADL software. After that, the data set will be divided into training. So you can use any type of software as per your need and convenience. After that, the data set will be divided into training, testing and independent validation, also called external set data set. This is essential to run the machine learning algorithm. 3D SDF structure is used for the calculation of molecular descriptor as well as fingerprints. Further, to take only relevant features and to rule out the possibility of overfitting of the model, feature selection will be preferred. We already know what is the meaning of overfitting and underfitting. Feature selection. Feature selection is an important step to extract the most relevant feature and to remove irrelevant feature to help to achieve high accuracy of the developed model. Only trained set will be used in all the pre-processing steps, that is removal of constant features, removal of highly correlated features and normalization of feature selection to avoid leakage of information from test set to the developed model. You should not use training data while using testing data to test the machine learning model. Otherwise, you are diluting your machine learning model and it will be victim of overfitting or underfitting based on the leakage. So this help in avoiding overfitting and positively biased results on the test set. Feature selection will be done using support vector regression implemented using LIB-SVM. This is the parameter to control the number of support vector. We all know the meaning of support vector. Already we have studied in support vector machine type of machine learning algorithm. Another way for doing feature selection is with the help of recursive feature elimination and Boruta. So you can use any of the proprietary tool to go for feature selection and normalization. Finally, most relevant features out of numerous descriptors will be selected. So for example, you have thousands of descriptors to describe any particular set of data compound, but you will choose only those features which are signature or main features of particular anti-cancer drugs if you are going for machine learning model for cancer treatment prediction. This is followed by model fitting and optimization of hyperparameters with the help of 10-fold cross validation. 10-fold cross validation is a type of validation of the machine learning model after training. So what is the meaning of 10-fold cross validation? The 10-fold cross validation means it is used to develop the predictive model. In the 10-fold cross validation, training and testing is divided into 10 sets. Whatever data you have, you will make 10 clusters. You will divide data into 10. For example, you have 100 input molecule. You will make 10 groups, each of 10 input, that is 10 compounds. Initially, the 9 data sets are combined for training and the remaining 1 set for testing to finally calculate the model performance. What is performance? You have trained the machine learning model. Now you are testing the machine learning model using unknown data and you are checking the performance. What is the meaning of performance? Is the model giving output which is actual or ground truth? It's called model performance. Suppose you are showing the image or if you are showing the metformin molecular graph as input to the machine learning model, and if it is identifying, okay, this particular structure is or can be used for treatment of diabetes based on so and so machine learning training, then it is called actual result given by the machine learning model. And if it is okay, it is true, then it is ground truth. So machine learning output should be equal to actual output or ground truth. This is called the model performance. So you have to minimize the output difference compared to actual ground truth. Likewise, all the sets get a chance to become the testing set. However, the average performance of all the testing sets represent the overall performance of the model. Further, the performance of the developed model is cross-evaluated using the independent data set, which will not be used during training and testing. This is the last stage we are talking about. So what are various performance measure? What are the yardstick to measure the model performance? So various formula or technical terminologies are there to judge or to evaluate the performance, that is the output given by the machine learning model or group of machine learning model. So in this particular case study, we have recommended to go for 10-fold cross validation using external data set. So the performance of the developed model can be analyzed using number of statistical parameters, that is sensitivity, specificity, accuracy, Pearson's correlation coefficient, that is PCC, mean absolute error MAE, root mean absolute error RMAC, Matthews correlation coefficient MCC, the percentage of the inhibitors which are correctly predicted by the model is called sensitivity, while the percentage of non-inhibitors which are correctly predicted by the model is called specificity, and accuracy is defined as the measure of the proportion of the predicting the data points correctly. MCC measured the quality of binary classification. MCC value ranges from -1 to 1. If the value is closer to 1, that means the model is highly accurate and the predictions are positively correlated with the actual value, that is ground truth. Ground truth. If value is closer to -1, that means there is negative correlation between prediction and the actual value, and if the value is closer to 0, that means the developed model is equivalent to random selection of class label. These are the parameters for evaluating machine learning model. There are few more formula, for example cross entropy and others for the evaluation of the performance of the deep learning type of machine learning model. With this, we finish this short video. Thank you very much.